Hello everyone, I'm Amima Mallik. Today I'm going to talk about the recent advancement in the field of genetics. Under that, I have selected the topic CRISPR-Cas9 Unleashing the Genetic Revolution Through Precise Editing. CRISPR-Cas9 means using a special tool called CRISPR-Cas9 to make really accurate changes in our genetic instruction. Just like you can edit a story to make it better, scientists can now edit our genes to fix problems or make improvements. This exciting progress is like a revolution in understanding and changing how our bodies work at the tiniest level. So, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Clustered, this refers to the way these repetitive DNA sequences are grouped together in the genome. Regularly interspaced means the sequences are regularly spaced out in the genome like evenly spaced beads on a necklace. Short palindromic repeats stands for these are short DNA sequences that read the same forward and backward. They are like genetic palindromes. CRISPR is a naturally occurring system that bacteria use as an immune defense against viruses. Scientists have harnessed this system to create a powerful gene editing tool called CRISPR-Cas9, which can be used to make precise changes in the DNA sequences. So, while the full form might sound complex, CRISPR has become a revolutionary tool for genetic research and editing. Let us move on to the components of CRISPR-Cas9. First is the target DNA sequence, second, guide RNA, third, PAM site, fourth, Cas9 protein. So, the target DNA sequence in genetics, the target DNA is a specific piece of genetic information or sequence that scientists want to work with or change using tools like CRISPR-Cas9. Second is guide RNA. The guide RNA is a key player in the CRISPR-Cas9 gene editing system. Think of a gRNA as a map or a GPS that guides the gene editing tool. Cas9 to the exact spot where changes are needed in the DNA. Scientists use gRNA to find the specific sequence in the DNA that they want to edit. Third is PAM site. The PAM site stands for Protospacer Adjacent Motif. It is a critical part of the CRISPR-Cas9 gene editing system. Imagine you are an editor trying to correct a mistake in a sentence. The mistake is like a part of a DNA you want to change using CRISPR-Cas9. But how do you tell the editing tool where exactly you have to make the changes? That's where the PAM site comes in. The PAM site is like a little flag next to the mistake. It tells the CRISPR-Cas9 tool exactly where to begin its work. So the PAM site is super important because it helps to guide the CRISPR-Cas9 tool to the right spot in the DNA where changes need to be made. Fourth is Cas9 protein. As I mentioned earlier that Cas9 protein is a pair of molecular scissors that can cut DNA. Scientists can use this protein to precisely cut DNA at specific places. By using Cas9, scientists can change these instructions in a controlled manner, which opens up a world of possibilities for treating diseases and understanding how things work in our bodies. Let us move on to the mechanism of CRISPR-Cas9. So first is double-stranded DNA. So the double-stranded DNA has 4 nucleotides. As we all know that cytosine pairs up with guanine by 3 bonds, adenine pairs up with thymine by 2 bonds, and in case of RNA, adenine pairs up with uracil. Next is target DNA sequence. Target DNA, it is that mutated DNA where we have to make changes. It is that unwanted sequence of DNA which we want to remove from the DNA in order to make the DNA precise. So next is guide RNA. So now guide RNA comes into picture which guides the DNA where exactly the cutting or cleavation will take place. So next Cas9 protein comes into picture. As I've already mentioned earlier, what is the role of Cas9 protein? To cut the DNA from its specific site. In the cleavation of DNA step, the Cas9 protein will bind to the unwanted part of DNA and it will help to remove that part with precise editing technique. After the unwanted sequence of DNA is cleaved by the Cas9 protein, now we can add our desired DNA in the double-stranded DNA. So this was the whole mechanism and process of the CRISPR-Cas9. Now, let us see how gene editing was done before and how the new advancement has helped us in achieving errorless and easy way to do gene editing using CRISPR-Cas9. Talking about the genetic modification, earlier it was complex laborious techniques like restriction enzymes and talents were used for genetic editing. Talens is transcription activator like effector nucleases. It is an artificial restriction enzyme that have ability to cut DNA at the point of contact with a series of nucleotides. Now, it has enabled precise and efficient gene using guide RNAs and the Cas9 enzyme. Talking about the efficiency, earlier it was time consuming and often had limitations in targeting specific sequences. Now, editing became more accurate, quicker, and efficient with the ability to target specific sequence. Next is accessibility. Genetic modification was challenging and expensive. Now, CRISPR-Cas9 made gene editing more accessible and cost effective. Talking about the application, earlier there was limited scope in medicine, agriculture, and research due to technical constraints. Now, it has enabled diverse applications in medicine, agriculture, biotechnology, and research transforming these fields. Now, let us move on to example and uses of CRISPR-Cas9. In medicine, it has helped in gene therapy, cancer research, and HIV research. In agriculture, it has helped crop improvement and livestock production. In biotechnology, it has helped drug development and bioproduction. In the research field, it has helped functional genomics, ethical considerations. It has helped human embryo editing. In human health, it has helped in malaria eradication. Overall, we conclude the topic that CRISPR-Cas9 impact on human health, disease eradication, and species modification is vast, striking. A balance between innovation and responsible use remains a challenge. It holds a pivotal role in genetic editing studies. Hence, CRISPR-Cas9 represents a groundbreaking advancement in the field of genetic engineering and gene editing. Below are the references mentioned. Thank you.